So do you remember that earlier on you have learned about how changes in the gene, okay, due to mutation, that means change in base sequence of gene, mutation, meaning change in base sequence in the gene, okay, in the your gene is your DNA, mutation in your DNA, okay, will lead to change in base sequence in the mRNA, which will lead to change in the amino acid sequence, right? In the polypeptide chain. And therefore, it will lead to change in the function. And therefore, it will lead to change in the phenotype. So we have looked at this in human, correct? Earlier on when we learned this topic. So today we will continue to learn this principle, but we apply it to the plant. So when there's a change in the base sequence of a gene in the plant, how does it affect the phenotype of the plant? Okay. So in the previous lessons, we have looked at how changes in the base sequence of certain gene in human will eventually lead to change in phenotype, and those kind of phenotypes are actually genetic diseases, because those diseases or illnesses are due to the changes in the gene. Okay, so example, the phenotypes are albinism, sickle cell anemia, hemophilia, and Huntington's diseases. And then in order to learn that, we also show the relationship, how the change in which gene that code for which product will lead to what phenotype. So we look at 4 examples, I think. So TYR gene codes for thyroxine, any changes to it will lead to albinism. HBB gene code for beta chain of hemoglobin, any change in it might lead to sickle cell anemia. F8 gene, any mutation may lead to factor VIII cannot be produced, so it will lead to hemophilia, the blood cannot clot. Okay, HTT gene code for the protein called Huntington. So how to remember? You go to the jungle to hunt for something. So hunting for something. Hunting for what? Hunting for tin. I don't know why I go there jungle hunt for tin, but it's Huntington. Huntington. Okay. So mutation to that change causes changes to this protein and leads to Huntington's disease. And the mutation leads to a dominant allele in this case here. Okay, so that one you can go back to revise. Now today we will learn also mutation that leads to changes in the gene that code for the height of a plant, how tall the plant will grow. Okay, so the learning outcome says that candidates should be able to explain the role of gibberellin in stem elongation. So how gibberellin can affect stem elongation, so explain it. Okay. So of course, then we have to look at how gibberellin causes stem elongation. And when mutation occurs, it causes gibberellin to change shape. And when it changes shape, it can no longer cause cause stem elongation. Okay, so let LE be represent a dominant allele that code for stem elongation. Okay, so it codes for a functional enzyme needed for stem elongation. So this is dominant allele. Let small le be a recessive allele, which codes for non-functional enzyme, because it changed in base sequence. The enzyme produces different shape and is non-functional. So therefore, the stem cannot elongate. Okay. So before I proceed, just a general information. Now, when we write human gene, we always use a 3-letter abbreviation like this one here. 3-letter abbreviation except for F8 to represent factor VIII. Okay. Now, can you see all of them are italicized in italic? So every time it's italicized, right? When you write using your handwriting, you need to underline it. So when you write, you have to write TYR underline. Now, for plant genes, you can see just now the plant genes, right? Usually, traditionally, plant genes have been given 2-letter abbreviation, like just now the LE. But it's also in italic. So when you write, you also need to underline them. Okay. Okay, now we proceed to look at gibberellin. Okay, for a normal allele and for a mutated allele. Okay, now the normal allele is dominant allele, big LE. So this dominant allele code for an enzyme for the last step of the pathway for GA synthesis. Remember, a pathway is made up of series of reactions, just like aerobic respiration, photosynthesis is actually a pathway made up of many, many steps. And every step, you got one enzyme coded by one gene, right? So this LE allele, okay, code for an enzyme for the final step of GA synthesis pathway. So once you got this enzyme, you will convert GA into active form of GA. So this GA will lead to cell division and cell elongation. So therefore, the stem will grow taller and longer. Okay. Now, what happened to the recessive allele, LE? Recessive allele, LE is a result of mutation. So there's a mutation in certain part of this gene leading to a different base sequence, so it's a different allele, right? So because of the mutation, the enzyme that is produced from here changes shape. So this enzyme is non-functional, it cannot function because the shape has changed. So therefore, this enzyme cannot catalyze this last step of the GA synthesis pathway. So therefore, it cannot convert inactive GA to active GA. So the GA remains inactive. So it cannot activate cell division and cell elongation. So there will be no cell division and cell elongation. So the plant remains stunted. Okay, the plant remains stunted. So here you got the here you got this arrow, right? So this arrow leads to tall plant. Okay, and this tall plant is coded by LE, LE, or LE, LE. So it's either homozygous dominant or heterozygous dominant. Whereas for this one, it leads to a dwarf plant. Remember, dwarf plant is very short. So this is coded by the homozygous recessive. Okay, in italic. Okay, in italic. Okay. One more information I want to show you is this one. So what happened here is that this mutation will cause alanine to be synthesized, alanine to be added into the polypeptide chain instead of threonine. Okay, so when they synthesize this protein, they use alanine instead of threonine at a certain region of the polypeptide. So where is this region? This region is near to the active site. So near to the active site, the polypeptide chain, let's say this protein, right? This polypeptide chain to form the protein. Okay, let's say this is active site, this region here. So let's say this particular amino acid here, let's say amino acid number 4, in the normal allele is threonine. Because of mutation, it becomes alanine instead of threonine. So because amino acid has changed, so the overall shape of the protein will also change. So it cannot function. So this is additional information from the textbook. Okay. So now, how do we measure stem elongation in the plant? It is measured by measuring the changes in the internode. So what is internode? Now you see node. Node is the knot or the joint in the plant at which the leaf arises. So you see, this is one node here because the leaf arises from the stem. This is another node because the leaf arises from the stem. Okay, so when you want to measure the rate of elongation of the stem, you measure the rate of elongation of the internode, like this diagram here. Okay, now there's some mistake in this part here, so I need to save this. Save. Okay. So you see, this is initial length. Then you allow the plant to grow. Then after that, grow for maybe 1 month. So after 1 month, you measure the length. So this final length. So then you can find out what is the rate of growth, right? The rate of growth is actually the rate of LF minus LI, okay, divided by T. So I should put like this, right? LF, LI divided by T. So that's the rate. Okay, so this is how you calculate the rate. So what you have learned is the effects of mutant alleles on the plant phenotype. And the example we use is the height of a plant. So learning outcome says that candidates should be able to explain the role of gibberellin in stem elongation. Okay, that means it will stimulate, it will activate cell division and cell elongation of stem. Okay, so what does LE code for and what does small le code for? Okay, so that's all for this lesson for this particular topic.